After studying this module, you shall be able to know the DNA microarray technology, learn the principle and procedure of DNA microarray techniques, differentiate between the types of microarrays and their technical aspects, and understand the uses of microarray technology. The genome of human beings contains approximately 30 to 40,000 genes which control the genetic traits like height, skin, hair and eye color and many more. At any given instance, cells in our body have variety of genetic combinations expressed or repressed, which in turn provides a specialization to that cell. For example, the cells in retina are photosensitive and are involved in vision, while some special cells in stomach produce enzymes that help in the route of digestion. Considering the number of specialized cells and tissues present in an organism and its complexity in combination to form the various organ systems, it would be a colossal challenge to identify and research on genes applying earlier techniques. The DNA microarray technology enables scientists to investigate expression of a number of genes rapidly in an integrated experiment in an efficient manner. In a microarray experimentation, RNA is isolated from a particular cell population or tissue and is hybridized to the complete genome complement of system. A microarray chip is constructed using several DNA samples of known amount. The quantity of mRNA hybridized on the microarray chip specifies the expression level of several genes. This data is taken and a gene expression profile is obtained for a particular type of cell. What is a microarray? A cDNA or DNA array is an ordered arrangement of samples where comparing of known and unknown DNA samples is carried out according to the base pairing rules. Microarray analysis comprises cell lysis, isolation of DNA or RNA, that is its genetic contents, and identification of all the genes that are expressed in that specific cell and of specific temporal stage. A microarray experiment can be achieved using microplates called chips. The size of sample spots is less than 200 microns in diameter and generally comprises of thousands of spots. Thousands of spotted samples of known identity called probes are fixed on a microarray chip, which is a solid support, for example, a glass slide or a silicon based chips or nylon or nitrocellulose membrane that helps in localization of an expressed gene. These spots can be DNA, cDNA or oligonucleotides. These are hybridized based on the complementary binding with the not known sequences, thereby allowing simultaneous analysis for genetic expression and gene detection. A single microarray experiment using DNA chip can deliver information on thousands of genes concurrently. A DNA microarray or a biochip is a microscopic consortium of DNA that are fixed or immobilized on a solid surface, which is used to quantify the genetic expression levels of all genes simultaneously. Each spot comprises of a particular DNA sequence called probe in picomolar concentration. A probe can be a small but specific sequence of a gene that is hybridized to a cDNA or cRNA also called antisense RNA sample, under an extremely controlled environment. The probe target DNA hybridization is then quantitatively detected by fluorescent signal from target labeled with fluorophore or silver or chemiluminescence tags to regulate the comparative concentration of mRNA or DNA sequences in the target. The principle underlying the technique of microarray is complementary hybridization among two DNA strands. The property of complementarity among nucleic acid sequences allows a specific pairing by creation of hydrogen bonds between complementary nucleotide base pairs. High degree of complementary base pairs in a sequence ensures strong bonding between the strands that resist the washing off, while non-specific binding sequences are removed using surfactants. Fluorescent labeled target sequences bound to a probe produce a signal according to the hybridization conditions. Total strength of the signal from a spot depends on the quantity of target nucleic acid bound to the probe present on that spot. Microarray apply comparative quantitation where the concentration of a spot or a feature is associated against the concentration of similar feature under a diverse condition. The individuality of the feature is defined by its location on the microchip. Types of microarrays. Depending upon the immobilized sample used to construct arrays and the information collected, the microarray experiments can be categorized in three ways. First, microarray for expression analysis. Second, microarray for mutation analysis, and third, microarray for comparative genomic hybridization. Microarray for expression analysis. As the name suggests, this type of microarrays use dioligonucleotides representing all genes. The experimental sample contains cDNA from both the normal as well as the pathological or diseased tissues. Thus, expression pattern is then compared to the expression pattern of a gene responsible for a disease. Spots with higher intensity are indicative of overexpression of genes in a diseased condition. Microarray for mutation analysis. This type of microarray experiment requires the application of genomic DNA. The genes might differ from each other by merely a single nucleotide base, commonly called single nucleotide polymorphism or SNP. Microarrays in the case locate the subtle differences in the nucleotide sequences and hence screen the mutations in the genome. Comparative genomic hybridization. This reveals information about the increase or decrease in the important chromosomal fragments of genomes being compared. These fractions may contain the critical genes that may play a significant role in progression of a disease or disorder such as cancer. However, based on analysis type, microarray includes DNA microarrays, which include cDNA microarrays, oligonucleotide microarrays, BAC microarrays, and SNP microarrays. chips for surveillance of microRNA populations, protein microarrays, peptide microarrays for detailed analysis or optimization of protein-protein interactions, tissue microarrays, cellular microarrays, also known as transfection microarrays, chemical compound microarrays, antibody microarrays, carbohydrate arrays, phenotype microarrays, reverse phase protein microarrays, microarrays of lysate or serum, and interferometric reflectance imaging sensor or IRIS. Microarray experiment. A microarray experiment has several steps. First, the two DNA samples directed for comparison are collected and checked for purity. This involves isolation of particular cells or tissues from treated sample and untreated sample or from disease versus normal. Second, the next step is isolation of nucleic acid or acids of interest, which can be RNA for expression profiling, DNA for comparative hybridization, or DNA RNA bound to specific target protein, which is immunoprecipitated for epigenetic or regulation studies. Third, the purified nucleic acid fraction is analyzed for integrity, quality and quantity to exclude the presence of any degraded nucleic acids in the sample by gel electrophoresis or a bioanalyzer. Further, sufficient quantity of RNA must be present in sample to proceed with the experiment. Fourth, the labeled product is then synthesized via RT-PCR method. A fluorescent label is added either during the reverse transcription or after the amplification. The labeling can be direct or indirect. For two channel arrays, the coupling stage is pre-hybridization using aminoallyl uridine triphosphate and NHS amino reactive dyes such as cyanine dyes. For single channel arrays, the coupling stage occurs post hybridization using biotin and labeled streptavidin. The modified nucleotides are added enzymatically in a ratio lesser than normal nucleotide. The DNA is purified using hydroxypeptide column. The aminoallyl group is an amine group on a long linker attached to the nucleobase, which reacts with a reactive dye. A dye flip can be performed on a duplicate sample to remove any dye effects in two channel experiments. Here a second slide is used with the label swapped and the sample that are labeled with CY3 in the first slide is now labeled with CY5 and vice versa. Fifth, the labeled samples are mixed with hybridization buffer containing STS, SSC, dextran sulfate, a blocking agent such as salmon sperm or calf thymus DNA, Denhart's solution. Sixth, the mixture is denatured and added to pinholes in the microarray. Microarray is hybridized overnight either in a hybridization oven where the microarray is mixed by rotation or in a mixer where the microarray is mixed by alternating pressure at the pinholes. Following hybridization, all non-specific binding is washed off using washing buffer containing STS and SSC. The microarray is dried and scanned by a laser based microarray scanner that uses a laser to excite the fluorescent dye and measures the intensity of emission as signal with a detector. The image is superimposed with template and the intensities of each feature are quantified. The raw data generated as signal intensity is normalized by subtracting background intensity and scale to make intensities of the features of the two channels equal. Normalization can be done by determining intensities of a reference gene to calculate the t-value for all of the intensities. There are more sophisticated normalization methods such as Z-ratio, regression and RMA, which is robust multi-chip analysis from the Affymetrix chips. Data analysis. Data analysis involves grinding, spot identification of the scanned image, removal of poor quality and low intensity features, subtraction of background fluorescence, determination of spot intensities and intensity ratios, visualization of data and log transformation of ratios, global or local normalization of intensity ratios and segmentation into different copy number regions using step detection algorithms. This is coupled with analytic approach based on available information and hypotheses underlying the experiment. Finally, statistical methods based on gene networks and gene interactions are applied. Microarray data may require further downstream processing to minimize the dimensionality of data in order to achieve better understanding and more focused analysis. Applications of microarrays. The technique of microarray has a plethora of applications in the arena of cell and molecular biology, recombinant DNA technology, genetics, disease diagnostics, etc. Some of the major applications of this technique are gene discovery. DNA microarray experimentations can be useful in recognition of undiscovered genes, understanding their molecular functioning and expression levels of genes under different conditions. Diagnosis of diseases. DNA microarray enables enhanced understanding of molecular bases of diseases such as heart disease, mental illness, infectious disease and especially cancer, where with present advancement in the area it might be possible to classify the forms of cancer based on the pattern of gene activity in the tumor cells. This will assist the pharmaceutical community to grow drugs that are more effective, as the handling policies will be directed straight to the particular form of cancer. Discovery of new drugs. Microarray technology has extensive application in pharmacogenomics. Relative examination of the gene expression from a diseased and an ordinary cell will assist the recognition of the biochemical composition of the proteins produced by the diseased genes, which can further help in recognizing new targets for drugs which combat with these proteins and reduce their effect. Toxicological research. Microarray technology can enable to study and analyze the influence of toxins on the cells and their progeny. Toxicogenomics forms correlation between reactions to toxicants and the variations in the genetic profiles of the cells reacted to such toxicants either naturally or by human intervention. Now, let us summarize what we have learnt in this module. A DNA microarray or DNA chip or biochip is the assortment of microscopic DNA spots joined to a solid area. Microarrays are applied to quantitatively know the expression levels of several genes concurrently or to genotype numerous places of a genome. Microarrays experiments are complex and are analyzed by the help of specialized softwares. Analysis includes normalization and expression level analysis. The technique of microarray has a plethora of applications in the arena of cell and molecular biology, recombinant DNA technology, genetics, disease diagnostics, etc.